Hello. So today I'm going to present you with DMT, frying your brain during brain injury, though, and also only maybe. And you can see here they are sipping on the ayahuasca. So yeah, let us get right into it. So first of all, DMT does seem to be protective in rats. Although they did this experiment with neuroprotective anesthetics that were already shown to protect in similar conditions. Now, there's also the fact that um, the combination of psychedelics and dissociatives, at least according to one study, appears to be, it appears that the 5-HT2A agonists are neuroprotective against the toxicity induced by the dissociatives. So that's another thing that should be kept in mind there. So another thing before we really get into it, anesthesia has been tested for brain injury as well, and it failed clinical trials. So how your brain dies? Yes. Okay, so there is glutamate excitotoxicity. Glutamate increases by as much, or I've heard someone else told me that it's even more in certain cases, but the study that I cited brought up 80-fold for up to an hour after the brain injury. This predicts worse outcomes in humans. The more glutamate someone has, the more problematic the outcome. So there are also NMDA glutamate receptors. These are an excitotoxic mechanism. They are also a proplastic and learning mechanism, which is something to consider that since DMT is thought to enhance plasticity and learning. Now, there's dinorphins, which may play a role in brain injury as well. But this is a little bit complicated because they might also play a role in protecting the brain. And so it's not as simple as we would hope it to be. So dinorphins are the endogenous form of salvia, in a sense. They are the kappa opioid receptor agonist. And you might know that salvia is a crazy hallucinogen. Here we have they are an anti-convulsant mechanism. There's some research on epilepsy and dinorphins and seizures and dinorphins. And it seems to have an anti-convulsant effect through the kappa opioid receptor mechanism. And it can be protective and toxic, which seems to depend on the circumstance. One of those circumstances is glycine levels. We won't get too much into that, though. It also is protective against seizure-induced excitotoxic damages, which are thought to be induced by NMDA receptor mechanisms. So there's also this interesting part. So there's research on dinorphins impairing cognition. There's research that they tried to see if it might be playing a role in Alzheimer's and dementia. There's also stress-induced memory impairments were linked to dinorphin. Also, alcohol-related impairments, learning and memory impairments, were associated to dinorphin as well. It also seems to be anti-plastic, which that will come into play in a little bit as we look at how DMT might be pro-plastic. So what does DMT do, though? So it seems to improve learning and memory in animals. It induces neurogenesis in animals. There's also seems to be plasticity effects, at least if we are to go by the rest of the serotonin psychedelics and even serotonin itself. The main mechanisms of interest are the sigma-1 receptor, This one's often seen as a mysterious receptor mechanism. This one enhances NMDA receptor activity, which you can kind of see where that might be going. There's also 5-HT2A receptors, which seem to have mixed effects on NMDA receptors, although this is a little bit trickier. We'll get into that soon. There's also the 5-HT1A receptor, which this seems to suppress dynorphin activity. So what about the dinorphinergic exorcism? Dramatic, right? So by this I mean that what if the effects of psychedelics are to exorcise dynorphin from you? The reason I think that is, as you see this third bullet point, dynorphin seems to induce the sort of effects that psychedelics seem to treat. It's been implicated in PTSD, stress, dysphoric sensation, depression, addiction, and there's anxiety that's not listed here. But if you look at the article, that stuff is listed. So this is a bit hypothetical, but there is evidence. 5-HT1A receptor agonists suppress dynorphin increases related to dopamine mechanisms. So the D1 receptor seems to be what increases dynorphin in general from a lot of different mechanisms. So, for example, it is likely to underlie the increase of dynorphin observed with many drugs. So morphine, stimulants, amphetamine, cocaine, caffeine, MDMA, and so on. So 5-HT1A receptor agonists seem to suppress the dynorphin increase, and the drug in the study used to increase the dynorphin in that case was levodopa, which is kind of a general dopamine stimulant. So LSD was found to suppress the behavioral responses induced by kappa opioid receptor agonists. So that's quite interesting. That might be due to this 5-HT1A agonism, which LSD does bind to. DMT does as well. Dynorphin, I already said that. So while dynorphin seems to be an anticonvulsant, and so do other kappa opioid receptor agonists, the psychedelics don't seem to be. They seem to be pro-seizure effects that come from them. So there are some case studies that I've linked to in the article that corresponds to this project. There's cases that were usually high doses of psychedelics. There's also the case that full agonists at 5-HT2A seem to induce those kind of effects. So like N-bones, that's kind of a famous one where people seem to get like seizures or really bad effects or even die from taking it. And the thing that separates it mostly seems to be that it's a full agonist, whereas LSD and other psychedelics that are commonly used are only partial agonists, which they don't really peak as hard in a sense. Okay, so what about this? Glutamate enhancement. This is basically the problem that I think is at play here that makes this a risky candidate for treating strokes or brain damage. So before I get into that, the company that wants to study this in using DMT for brain injury, they specifically mention using it as people enter the ambulance. And that's kind of where I find it most problematic. I actually think it might enhance recovery if they just waited about an hour maybe, or even maybe even longer just to be sure. But let's continue on. So 5-HT2A receptors are thought to play a big role in the psychedelic effects, and it seems to be through enhanced glutamate release. And you can kind of contrast this with what dissociatives do. They block glutamate receptors, and they kind of numb you and detach you and make everything distant and more faded and less sensitive, basically. But it's a little bit more complicated. But so the psychedelics, on the other hand, seem to enhance glutamate activity. And so what we observe subjectively, at least what it seems to be, is enhanced visual acuity, enhanced sensitivity to emotion, to maybe even tactile sense, or enhanced maybe taste, enhanced kind of everything. It seems to be like people talk about this increased consciousness, right? That might be what is going on that level. Not to say that the effect actually is enhanced consciousness. That's debatable. That is for a different video. So this could potentiate, this effect of enhancing glutamate might potentiate glutamate-mediated brain damage, which seems to be one of the key players in brain injury. So that's pretty significant. Glutamate and NMDA receptors are pro-plastic and pro-learning. That's something to remember, which that might underlie the effect of psychedelics on plasticity and learning. So a nootropic, IDRA-21, enhances glutamate and brain damage as well. So that's something that's been observed in animal studies. That's despite the fact that it enhances cognition and seems to be pro-plastic as well. LSD potentiates this same mechanism that IDRA-21 potentiates, which is the AMPA receptor. And AMPA receptors and NMDA receptors, they both kind of play together in this very complicated role, and they both can lead to excitotoxic effects. So, yeah, a bit spooky. So, NMDA receptors. Serotonin promotes NMDA receptor activity in motoneurons in animals. Psychedelics promote NMDA receptor activity in the prefrontal cortex of animals as well. But it seems to be dose dependent in the case of psychedelics. Low doses seem to potentiate it, but high doses seem to inhibit it. And this is kind of a peculiar thing. I think it might maybe relate to whether or not maybe it has to do with the 5-HT1A receptor. I think it's a bit complicated, so I don't know how far we can speculate, but in the article it kind of goes into depth speculating if you're curious. So this is the mysterious sigma receptor. This receptor is thought to play a role in the neurogenic effects of DMT. Sigma-1, so it's an agonist. I should be clear about that. DMT is an agonist of sigma-1. Sigma-1 receptors potentiate NMDA receptors, so that's where it gets a little bit trickier. Sigma-1 agonists have been shown to be neuroprotective, but here's the catch. Some of those, maybe even all of them, I need to go back and check, but more than one of them at least were negative modulators of NMDA receptors, which is really significant. And we would also have to check if they're using things like anesthetics, again, because those block the NMDA receptor. But so yeah, I would be really careful in that case because that's a mechanism that might be neuroprotective on its own. Suppression of sigma-1 via CBD, interestingly enough, seems to be protective, and that protective effect was reversed by a sigma-1 agonist. So that there does not really suggest protection. And then a sigma-1 antagonist has also been shown to be protective in ischemia. and they mentioned that presumably via reduced NMDA receptor activity. So the fact that DMT is a sigma-1 agonist, that does not necessarily mean it's going to be protective in ischemia, especially since we've observed precisely the opposite. So here's where we get into a little bit of the subjective effects that you might find interesting. So we mentioned 5-HT1A suppresses dinorphin. 5-HT1A antagonists actually potentiate DMT, most strangely. Now that tells me that it might be possible that DMT is inducing dinorphinergic effects, and that by suppressing 1A, you're enhancing that other mechanism that might be inducing dinorphin effects. So in some sense, DMT might have overlaps with salvia. 1A antagonists also facilitate the NMDA receptor inhibition via serotonin itself. So now that's even another layer that gets spooky, because that means that blocking 1A seems to essentially make serotonin more like a dissociative, and perhaps by enhancing dinorphin and its dissociative mechanisms. So the next stage here is dopamine. So dopamine-induced increase of dinorphin seems to rely on the legendary D2 receptor. That's the one that people talk about in schizophrenia. So that's kind of interesting. And I have a whole dinorphin theory of hypothesis, I guess, of schizophrenia that I might make a video later. That's such a elaborate one, but I might do that at some point. But there is a post already out about that, and I'm working on more of them. So there are D1, D2 receptor heteromers, which are connected mechanisms, basically, and they are on dinorphin neurons. And in particularly what's strange is that they are in a high affinity state in schizophrenia and meth users. So that is quite striking. Like, basically it implies that there's this dinorphin enhancement mechanism in schizophrenics and meth users, so that might explain psychotic symptoms that might be occurring through kappa opioid or NMDA receptor inhibition mechanisms. So it might be like an enhanced ketamine or salvia-like mechanism going on in people with schizophrenia and meth users. So psychotic mechanisms may be driven. I already said that. Yes. Okay, so 5-HT2A receptors come into play here because they potentiate D2 receptors, which might facilitate dinorphin release by these mechanisms that we've just been outlining. So that means that the tripping from psychedelics, if it's mediated by 5-HT2A, part of this might be from not just glutamate release, but also dinorphin release, which would be a twist that is unexpected to me, at least. So, said that already. I should delete that bullet point. It's a duplicate. So the suppression of NMDA receptors could be related to dinorphin in all of the cases that we described already, like in the case of 5-HT2A receptors. So this is weird because this is where protective effects of DMT might depend on various receptor types. Like, should we be combining DMT with anesthesia always? Should we be combining it with 5-HT1A antagonist or agonist? We don't really know the answer to that. And just going back, let's have a remembrance for the dissociatives. They failed clinical trials. And so in a paper that talks about the failure of dissociatives, they mention that it might be due to the fact that the acute, it might be protective in the acute phase, like, for example, that first hour where brain damage is peaking via excitotoxic glutamate mechanisms. In the post-acute phase, though, it may actually impede recovery by suppressing NMDA receptors. It might be suppressing plasticity and potentially other things, maybe neurogenesis or something. We don't know about that yet, but the other thing about dissociatives, though, that it wasn't mentioned in that paper that I would be kind of curious about is they also increase glutamate release even though they're blocking certain glutamate receptors. So that totally complicates the situation. So like, if you look at trip reports, they've talked about like low doses of like DXM or maybe even ketamine, where they report kind of like sometimes enhanced visual acuity or sensitivity to senses or something like that. Perhaps that's the increased glutamate release, and then higher doses might become so inhibiting that it changes. I don't know. They also decrease GABA, which that can potentially be like pro-seizure and potentially neurotoxic. There have been observed paradoxical seizures, despite the fact that high NMDA receptor activity is linked to epileptic mechanisms and seizures. Some of the drugs, or even states with low NMDA receptor activity, have produced seizures as well. So this is not that simple. And it is never that simple. For example, consider the case of MDMA, ecstasy, Molly. There was a study where they tested at least 2 different doses where one of them was not neurotoxic, and that dose didn't produce any neuroplastic effects. Most strangely, maybe not even strangely, the plasticity enhancing dose was the neurotoxic dose. So, and it might be through enhanced NMDA receptor activity, which is both plastic and toxic. So it is almost never a clean, simple puzzle. Patients may appear to superficially recover if we give them DMT. That's another thing. So if it suppresses dynorphin or enhances learning or plasticity and memory mechanisms, remember those are also excitotoxic mechanisms. So yeah, that's really risky in my opinion. They might seem to improve before our eyes and actually be accelerating the damage they're doing to their brain. Like, you could imagine there's probably like some nootropics, like even that IDRA21, you could imagine like just blasting the dose on it and then observing your brain deteriorate alongside like greatly enhanced cognition or something like that. So we have to be careful. That said, DMT might be good. It increases during cardiac arrest in rats, it seems. So the great psychedelic scientist Nichols has pushed back on that. But if you look at the article that I wrote, you can, I talk about it in there. You can see the details of that. So there is research showing that DMT is protective against hypoxic brain damage via HIF1, which is hypoxia-inducible factor 1. So what's interesting about this that isn't really talked about all that much, but I try to bring it up, is that hypoxia disrupts monoamine oxidase. And think about it, this is an oxidase. It literally is an enzyme that works in the presence of oxygen. So if there is no presence of oxygen, then perhaps DMT will accumulate and induce this kind of HIF1 mechanism and be neuroprotective. And this might even suggest that there's an endogenous role in the case of hypoxia. And my recommendation, though, is perhaps we should use it mostly in the post-acute phase, maybe at least first. I mean, whatever, go experiment on dying humans if you want. But yeah. Thank you. You can see the full article in the link there's a link in the description. This is the website mad.science.blog. You can find it there. It's one of the recent articles. If it's not recent anymore, just type in DMT and you'll find it. It's obvious which article it is. It's the brain injury one. And yeah, so I hope this was interesting to you. Have a good day. Subscribe to the Patreon, please, so I am not poor. But yeah, so there are 12 Patreons. Shout out to all of you. You've literally saved me sometimes, even though it's not a crazy amount of money right now. But yeah, it actually is more useful than you would imagine. So yeah, you will gain access to exclusive content. There's some discussions there. There are new ideas that I don't post on the blog yet. There's trip reports about prescription legal ketamine, and my use of an EEG to record the data while using ketamine. So that gets kind of interesting. Yeah, so I hope you found this interesting, and that wraps it up for today. So I hope you guys are doing all right out there in the end of the world or whatever, the dystopian situation that we have on our hands. Try to keep hope. And remember, the Renaissance came after the plague. It might be a kind of cultural adaptive radiation. So just keep hope and wait it out, please.